Alzheimer's disease, the most common neurodegenerative disease. It eventually devastates cognition and one's ability to complete daily tasks. It is also the sixth leading cause of death in the United States. Despite decades of research, however, how it develops is still poorly understood, and current therapies are not effective. For a fresh perspective, our lab began researching whether gut bacteria might impact Alzheimer's. This connection sparked our interest because some of the risk factors for Alzheimer's can be causally controlled by the gut microbiome, a term I will use to refer to the bacterial life living in the intestines. We collected fecal samples from individuals with Alzheimer's and individuals of the same age and sex that had normal cognition. Interestingly, we found significant differences between the gut microbiomes of these two groups. This launched my project, a project to uncover whether specific bacteria and the small molecules they produce may influence Alzheimer's. One such small molecule is trimethylamine N-oxide, or TMAO. TMAO is produced from the dietary component choline, found in eggs, beef liver, nuts, and fish. While we need choline for important host functions, some bacteria are able to consume choline as a nutrient source for themselves. In the process, they produce TMAO, which then circulates in the host's bloodstream. The brain is richly supplied with blood, so we wondered if TMAO might impact Alzheimer's. Our lab found that TMAO is elevated in individuals with Alzheimer's, and that levels of TMAO correlate with Alzheimer's biomarkers. Going beyond correlation to causation, I am now using mice that model Alzheimer's to test TMAO's impact. First, I am generating bacterial communities that can and cannot produce TMAO and implanting these communities into the guts of mice that originally had no microbial life in or on them. To look at TMAO's impact in another way, I am supplementing another set of Alzheimer's mice with TMAO via their drinking water. Elucidating whether TMAO is causally linked with Alzheimer's will increase understanding of disease development and provide potential therapeutic targets, such as preventing bacteria from making TMAO. This increased understanding means so much when our loved ones could be more like the people they've always been, with less of an impact from Alzheimer's disease.